In this class, we can have a discussion on the importance of plant breeding, then what are the steps which are involved in plant breeding, and apart from that we can also have discussion on some of the examples wherein the varieties of plants have been developed in plant breeding to overcome the problems of maybe the pest or maybe some of the biofortification. All those things we will be able to understand in this particular session. Here, if you take the history of human civilization again, human being started domesticating animals for his food, and apart from that, he started growing some plants in a small field, small area. And the growing of these useful plants in that area is what we call it as agriculture, and those plants which are grown are called crops. And once he started growing these plants, most of the plants were available in the nature and they are called wild plants, wild variety also we can call it. And during the process of civilization, he started changing the plants through the crossing and all. Unknowingly started doing it during the process of civilization. And that resulted in some of the useful plants for them so that they can get the food which they want, or maybe to get cloth, or to get some of the other useful products which can be used by the human beings. And if you look at the history of India, as far as the availability of food is concerned, we know we face with the problem of population explosion. And immediately after the independence, we experienced severe drought situations and also severe shortage of food. So we were exporting so many food items, including wheat, rice, everything from the Western countries. That means we were not independent as far as our food requirement is concerned. We were depending on other nations. In this situation, we have father of Green Revolution and his team in India, Dr. Swaminathan and his team, to bring about Green Revolution, which paved the way for the enough amount of food yield in our country. So we were able to grow enough food stock so that we can feed each one in our country. So what made it possible for us to have this green revolution? It is not only in our country in the 1960s and all. So in the entire world, the process of green revolution started. The main reason for this one is plant breeding, and apart from that, using the scientific methods of farming, where they started using the fertilizers and maybe the machines they started using here, and in a large scale they started cultivating the fields. So the major contribution for the increase in yield is because of plant breeding. You know, in the natural way of reproduction, the crossing takes place between the female and also the male flowers. That is, the pollen grains from the anthers are going to get pollinated onto the stigma of the flower. That can be a cross pollination, and which results in the genetic variations. Because of the genetic variations which are happening, we may get a new variety with some advantages, and sometimes it may also result in some of the disadvantageous characteristics in those plants. Then what exactly is this plant breeding, which is also called hybridization? The one we get through this plant breeding or maybe hybridization is also called the hybrid. So what is that which we do in this? We are going to change the genome of the species during this plant breeding. So all the characteristics depend on the genes which are present in any organism's body. In plant breeding, it is the heart and science of changing the genetics of plant species in order to create desired plant types which are better suited for cultivation. In the sense, they are the ones which are giving us the better yields, better quality and all. So what is it which we look into whenever we go for plant breeding? So what are the traits, what are the characteristics we are looking for whenever we go for plant breeding so as to get a new variety which we call it as a hybrid. So we want to incorporate certain characteristics in them. We look for certain characteristics, certain traits whenever we go for plant breeding. So the first and foremost important characteristic, the trait which we look for is having best yield, the high yield. So we want to have a yield variety. Then secondly, better quality produce. So it is not only the quantity, high yield is concerned with the quantity. But here we are also concerned with the quality. Quality as far as the nutrient content is concerned. Quality with respect to the keeping status or keeping quality, you can say, which we also call it as shelf life. So this is the second important characteristic which is looked into whenever we try to develop a new trait. Then they should be able to tolerate the changes in the conditions in the environment. Being the agrarian country which depend on monsoons, we have to play with the changes in the monsoon. We have to subject ourselves to the fortunes of the monsoon. That's why here we need to have the plant varieties which have resistance or tolerance to the environmental stresses. That is, increased tolerance to environmental stresses, like maybe the water scarcity can be there, sometimes more water can also be there, or it can be the temperature, or it can be the salinity. Continued use of irrigation leads to salinity. So these are all the environmental stresses which are there, and they should be able to overcome this particular problem. And another important thing is the resistance to the pathogens. So there are so many pathogens from different groups of microbes, like maybe the bacteria, or maybe the viruses, or maybe the fungi, and also there are so many other worms which are affecting the plant yield. So they need to be resistant against these pathogens. And they should be tolerant to the insect pests. So there are so many insect pests which are damaging almost around 30% of the plants, or you can say the crops. So that's why these are all the important traits which they try to incorporate, which they look for whenever they go for plant breeding. You can just see the variety you can find with reference to the wheat and also other plants. In the plant breeding, we are going for such variety in this. And you see the result of Green Revolution. So it resulted in the increased irrigation facilities and also use of fertilizers, pesticides, and ultimately resulted in better yield. And also they started developing different breeds, or you can say the varieties of plants through this plant breeding. You can see the increase here. This is because of Green Revolution. And what are the steps which are involved in plant breeding? So mainly there are 5 steps which are involved. We'll go one by one. First, let us see the main steps which are involved. One is collection of variability. Secondly, evaluation and selection of parents. Cross hybridization among the selected parents. Fourthly, it is selection and testing of superior recombinants. Fifthly, testing, release and commercialization of the new cultivars. So what are these things then? So what is meant by collection of variability? So let us see one by one. If you look at the environment, we have variety of plants within the same species. If you take jowar, we have so many varieties of plants. If you take rice, we have n number of variety of rice. Like that, if you take any crop plant, we can find so many varieties of the particular crops. We have so many plants which are there in the wild, which we may not be using now, but definitely they will be having some characters, some traits which are useful. And there are some plants which are related to this particular species. So we may try to have those things. Like this, whatever the variety of crop seeds, those plant seeds which are available as far as that particular species of plant is concerned is going to be collected. And they are going to be thoroughly studied, and whatever the characteristics they are possessing are going to be documented. So documentation is one of the major thing which is required, because even though the crop yield may be less for a particular variety, but it may be very rich in, let me say, iron content in that, mineral content in that. There may be a variety which gives us very less yield, but very rich in some protein, like let me say beta carotene. So like that, we have certain varieties which have lot of advantages, lot of good traits, which is all because of the genes which are present here. That's why this collection and preservation of all the different varieties, which includes the wild varieties and the cultivated varieties, then the species and the relatives of the cultivated species, is what we call it as collection of variability, which is also called germplasm collection, which is called germplasm collection. You can look at this one. So we have variety of jowar seeds which have been collected in this. Like that, you can take any, it can be wheat, it can be rice. So you can see variety, that we call it as germplasm collection. It is, which is nothing but collection of variability. Once we collect the variability, we have to go for, I told you, so in the collection of variability, in the first step, we are going to document all the traits which are possessed by the particular variety of that particular plant. Then we have to find out whether they possess those traits or not. That's what we call it as evaluation. And depending on our requirement, what is that which we require? So whether it is the yield which we want to increase, or we want to have a variety which is able to be grown in even the saline soil with the higher salinity, or maybe we want to incorporate more amount of some protein. So like that, based on that requirement, so that particular varieties are going to be evaluated for such characteristics. So that is what we mean by evaluation, where evaluation is done to identify the plants with the desirable characters, the characters we want. Then these selected parents are going to be multiplied because we want to use it for crossing. So they are going to be increased in number, and then that particular process is what we call it as hybridization, where we are going to multiply this one. So normally the cell pollination will be done for this. Then after that, once we get, so we select the two different varieties with the desired traits. Quite naturally, they will be having some undesired traits also. And these two selected parents with the desired traits, let me say, one is with high yield, another one we have selected for, let me say, with high resistance to the pathogens. Then both these plants are going to be crossed. That we call it as cross hybridization, and these selected parents are going to be crossed here, and by crossing that one we are going to combine both of them. So as I told you, we have taken, let me say, in the first case, in the first parent we have taken the one with good yield. In the second parent we have taken the one with good resistance to the pathogen. So both are going to combine their traits genetically. So when they combine the traits, so we are going to get many plants in them. It is not that we will be going to get only the plants with both combinations, with good yield and also resistance to the diseases, because some recessive traits may also get exposed. So we will be getting so many plants with the different combinations. That's why we get the hybrids in this case. When we go for hybridization, the next step which we are going to have becomes very crucial for the selection. So in the fourth step, we are going for selection and testing for superior recombinants. So as I told you, we want to have a variety with both characteristics, with good yield and also resistance to the pathogens. So for that, what have we done? So we have so many plants. So we have got so many seeds, which some of them, very few of them, out of thousands of seeds, maybe 2-3 will be having the combination which we want. That combination which is desired by us, which is required by us, is what we call it as the one with the superior recombinants, because the recombination has happened here. So mixing of traits has happened in this case. So that is what is done in the fourth step, that is selection. We have to select it, which is a very, very important step in the process of plant breeding. If we are not following it properly, if we are not selecting properly, then it may not give us desired results. So that's why it has to be done scientifically. You can see the selection process is crucial to the success of the breeding objective and requires careful scientific evaluation of the progeny. And these are quite naturally, once we select them, we have to increase them in number. Because I told you, out of thousands of seeds we have got, maybe 3 or 5 may be having the superior recombinants. So for that, we have to go for multiplication, which is done by self-pollination. Then why should we go for self-pollination here? Why can't we go for cross-pollination? So if you are going for cross-pollination, those recombinants may lose the traits. The traits can be lost also. That's why you have to go for self-pollination, so that the characters will not segregate. They should not be segregated. We want to preserve those recombinant characters. That's why we have to go for self-pollination. Once we select and test for the good traits, so then we are increasing in the number, and then in the field, small fields we are going to use here under the supervision of the researchers. And these researchers, what do you call those researchers who are involved in plant breeding? What is the name given to the researchers, the scientists who are involved in the plant breeding? They are called plant breeders. They are called plant breeders. And these plant breeders will be taking these seeds or plants which are having the good recombinants, and that is the one with the desired traits, and they will be growing in the smaller areas, smaller fields, and test for the traits. And there, once they test it, they use it in a bigger fields with the help of the farmers. They will be selecting some farmers and they will be helping in using it for them for testing here and in a bigger field. Then they increase the amount by growing them in larger fields, and they go for, once they test it properly, once the testing is successful, once they feel that the hybrid which you have got is having the new traits where the recombinants are there. So in the example which I have taken, so all the seeds should be having good yield as well as the good resistance against the pathogens. And such ones are going to be selected, and they are going to be grown in big way with the help of farmers, and they are going to be released for the use by farmers, which we call it as commercialization. So that is going to be used for commercialization of new cultivars, the farmers in this case here. So like that, in the new locations, new areas, they are going to grow. So these are the 5 different steps which are involved in the process of plant breeding. And each step is very, very important. And if we fail at any one step, that plant breeding process is going to be a total failure. You can see the farmer's field where normally they try to use it in a larger scale. So this is about the plant breeding. This is one of the variety which has been developed. We have so many varieties which have been developed here. So Vivek maize hybrid 27 is one of the maize variety which has been developed here in our country. So like this plant breeding resulted in spurt in the production of many crop plants, especially the staple food items like wheat and also rice production. You know, wheat and rice are the 2 major staple food sources. So earlier, if you take wheat and also rice, both if you take, they were growing the wheat and also rice with more height, and they were taking many days, 160 days, 180 days sometimes, that is almost 6 months. So 5 months to 6 months were the lifespan of these wheat and also rice plants. And because of the research which was done here, they started developing semi-dwarf variety. Semi-dwarf variety. You can just have a look at this one. So this is the normal variety which they were growing earlier. This is the normal variety of rice, and this is the normal variety of wheat. This is the normal variety. You can look at the height. We have many varieties which were growing for about 6 feet earlier, before 1950s and all. Then later on, they started growing the medium height plants. You can see a medium height here. This is medium height here. Okay, so they are said to be semi-dwarf varieties. What are they called? They are called semi-dwarf varieties. They started growing. So these were the normal ones with more height, and this is one of the important thing which was done in this case, because they are having their own advantages. So there was decrease in the lifespan, maybe 120 days to 130 days in many cases. There are now, we have some varieties which can be used, which can be harvested within around 100 days. Such is the case now. So these semi-dwarf varieties have been developed here. And the credit for developing these semi-dwarf varieties of wheat and also rice goes to some of the prestigious international research centers. So it is ICWMI. This is the center you can see, which is in Mexico, International Center for Wheat and Maize Improvement in Mexico. This is the one which is involved in, even today it is involved in the development of wheat varieties. It was because of the great scientist, a Nobel laureate, was recognized and given Nobel Prize in 1970 for his contribution in developing the semi-dwarf varieties of wheat. He developed, it is Dr. Norman E. Borlaug, you can see him, great Dr. Norman E. Borlaug is the man who steered the process of green revolution in the world. He is the man who started developing semi-dwarf varieties of wheat. And so he started developing semi-dwarf varieties with shorter lifespan and also very high yield. So that is the one which is there. And we can see two varieties which are developed in our country. So now we have the wheat variety by name Sonalika. So it is one thing. It is also semi-dwarf variety. It is called Sonalika. You can see the breeder who is involved. He is the scientist. You see how they work here in the fields. You can see the name and all is given here. It is for easy identification, and this is the research field where they have grown this semi-dwarf variety. Here it is an example of Sonalika we are having. There are two important varieties which were developed in India at that time. One is Sonalika, and this is another variety called Kalland Sona. So two important varieties of wheat. One is, you can see Sonalika, and secondly Kalland Sona. So they were developed they were used in our country to improve the yield. Then apart from that, you can see what is the advantage in this case here. So they had better yield, and you can see Sonalika was developed in 1967 and Kalland Sona was developed in 1970. And we had other earlier varieties which were developed in 1965 also. But the yield potential you can see here. So it is 33.7 quintal per hectare for 2.5 acre. It is 36.0 quintal per hectare in the case of C306. But look at this Sonalika. So they had 45.5 quintals per hectare, and Kalland Sona 46 quintal per hectare. You can say almost 10 to 12 quintals increase in the product. And secondly, they are affected by many diseases. Normally the wheat was affected by this fungal disease, fungi, and so they got resistance. We will see the different examples later on anyway. So they had resistance against such diseases here. And we have another important food grain apart from wheat, which is one of the major food that we call it as staple food. It is rice. So in the world we have an International Rice Research Institute which is located in Philippines. You can look at the picture of this International Rice Research Institute in Philippines. So they developed, for the first time, they developed a variety called IR8. It is called IR8. You can see this is another semi-dwarf variety of rice, just like the wheat. I have shown you already, just like the wheat, they developed semi-dwarf variety of rice also. And this variety of rice, as I told you, they had less maturation period. And they developed in this institute, they developed another variety called Taichung Native 1. And some of the Indian varieties have been developed in our country, like Ratna and also Jaya. So they are the semi-dwarf varieties of rice and which have lot of productivity. The yield is very good. Jaya and also Ratna now we have so many varieties of rice, including Sona Masuri and all. You can see the year of notification and also the duration. So Taichung 65, 130 to 135 days. Now it is almost 120 to 130 days is the average maturation period as far as the rice is concerned. Now Jaya 130 days you can see. Now we have some Sona varieties which are going to mature within 120 days, around 100 to 120 days. That is the thing we are having. And another important thing which has been done. So we have the sugarcane variety, Sacharam barberi. This is the variety which was grown in North India. The problem with this barberi variety was it was not giving us good yield. The sugar content was very less in that. Wherein we have another variety, Sacharam officinarum, that is the variety which was grown in South India. This has very good sugar yield, and also the yield per hectare is also very good compared to the variety which was grown in North India. But the problem with this, so then they could have taken this Sacharam officinarum to the parts of North India, then they could have grown, But another important problem we face here, the adaptability. This Sacharam officinarum was unable to grow in the conditions of North India. So for that, what they did, our plant breeders did, so they crossed the Sacharam barberi and also Sacharam officinarum. And we got a new variety out of this with high yield, thick stems. You can see the thick stems here, then high sugar content and ability to grow in North India. So like this, hybridization resulted in a new variety which can be grown in North India with high sugar content. The problem with this Sacharam barberi was it was able to grow in North India, but sugar content was very less. So that particular problem was solved by developing a new variety like this. Then we have been able to develop so many millets. So these millets, many varieties of millets have been developed through this plant breeding. Millets are very good sources of food which are rich in fiber and also the minerals. And now these millets are the ones which are preferred a lot when compared to the wheat and also rice. And nowadays we are having so many varieties of millets which are called Siridhanyagadu. So that is one which we are having. Then we have to see how plant breeding is useful for disease resistance. This, as we know, the plants are affected by many diseases. You can see the fungal diseases like brown rust in the case of wheat. This we have shown in, this is the, against this only we have developed Kalyan Sona and also Sonalika. Then red rot of sugarcane, these are the fungal diseases caused in some of the plants. We have so many fungal diseases caused by most of the plants here. And you can see late blight of potato. So you might have seen such plants where the leaves also, as well as the tuber which we get, is also going to get affected here. Then we have some of the bacterial diseases like black rot of crucifers. Like this, we have viral diseases caused by viruses like tobacco mosaic disease, then tobacco mosaic disease, then tomato mosaic disease. You can see the tomato mosaic disease. Then we also have, we can see, apart from that, we have so many other viral diseases which are going to be there. And what is to be done for that? We have to develop the new varieties which are able to resist such diseases here. So how exactly we are going to develop that one? Again, it is through the breeding. So that how exactly we are going to develop through breeding and all, we'll see in the next class.